of my work in butterflies, in color patterns, trying to connect basically genotype to phenotype. I'm using butterflies because I think it's a beautiful system to study morphological evolution. And especially in the Nepalese butterflies, there's a ground plan that has been described since the 1920s. That it comes basically classify different elements in the wings in parallel elements. So we have basal and discal elements, the central symmetry system, the area where the eye spots are, and the margin, all the elements in the margin of the wings. We have at this point observed that there's this gene called wing A that is very important, or is basically associated with different elements of this lymphatic ground plan. So this is a really good marker for this ancestral expression pattern. When we knock out this gene and the butterflies using CRISPR-Cas9, we see drastic changes in the phenotype, and it's basically affecting different elements of this lymphatic ground plan. Now, there are butterflies that have diverged from this ground plan. Some of them seem to be like a little diverse, and some are like very in the stream. And when we look like the expression pattern, the mRNA expression in imaginary disc of the caterpillars, we see like a very divergent expression compared to the ancestral expression that I showed you before. And when we knock out the gene, again, CRISPR, we observe like drastic changes again in the butterflies. Another really cool aspect of this gene is that it has been mapped in Heliconius butterflies and in Limelitis butterflies here as being very important or behind the adaptive radiation of this species. So now the question I'm trying to understand and going behind is what's going on, or what is happening that is changing the expression pattern so drastically in these different species in a gene that seems to be very key in the evolution of the color patterns. The hypothesis I'm going behind is trying to understand the cis-regulation behind, basically trying to identify the promoters, the enhancers and repressors that are interacting with this gene and evolving this new expression pattern. To do that, I have collected imaginary discs when the wing A is expressed, but also collect the tissue from the heads. With this, I build ataxic profiles. And ataxic is a method that allows me to identify regions of the DNA that are without nucleosomes. So this is naked DNA that is what I call open for business to interact with transcription factors to activate or repress gene function. So when we sequence, we align the sequence and then they form these peaks, and those peaks are basically open chromatin regions and are putative regulatory elements. So the data looks, what you're going to see, it looks like that. So we have forewing, hindwing, and what I'm looking for are regions that are differentially accessible between the wing tissue and the heads. Now, this is the wing locus, and it's a landmark. These are the exons of the wing, so we have a huge intron, and then there's multiple peaks that are differentially accessible between the wings and the head, and this is showing like a complex regulatory landscape for this gene, and it's the same case for all the other species I looked so far. Now, how to start to untangle this? There are a lot of peaks to go through. So start to think about conservation. So if we have an ancestral phenotype and maybe conserved peaks that are in conserved regions could be important. But then we have like different degrees of conservation, like this peak in a region that has sequence conservation, but could be between two species, or there are peaks that are in non-conservation at all. So having this in mind, I thinking in three questions. First, is like the sequence conservation associated with the function conservation. So is this bar I'm showing here underneath is like sequence conservation against an ancestral, against the Vanessa butterfly, this ancestral part. So is the function being conserved or is varying? can novel functions evolve from conserved regions? So for example, this region seems to be very well conserved in all the species, but the function seems to be divergent. So what's going on there? Or can we see the novel expression patterns evolving from the novel sequences? To answer this, I'm using CRISPR-Cas9, and so I inject X to disrupt these regulatory putative elements. And what we usually obtain is like mosaics. When we are doing, or what I'm showing you here today, it's only F0. I don't have lines for these elements yet. So what we see is, for example, this is the knockout of the optic gene that is related with the red pattern. So this is wild type, and this is a mutant, and this is a close-up. So this is the kind of things we are observing, just mosaic clones inside the genes. Okay, let's click some of the results. So this is the lady painted butterfly. This is the wild type phenotype, and I want you to focus on the biggest eye spot here in this hind wing, and you will see all these fine, beautiful decorations. When we knock out the gene, all these decorations go away. So this is related with the central symmetry system of that lymphatic ground plan. Now, when I knock out this gene, that seems to be conserved with other species, we observe the same effect. So we have a regulatory element that's directly associated with the central symmetry system in this case. So peaks on conserved DNA regions drive conserved expression patterns. Now, what about these butterflies that have more diverse phenotype? Again, just focusing this area, we see these silver spots in the wild type. When we remove the wing A gene, all those spots go away. And when we remove this regulatory element that is very diverse, we'll see the spots stand completely, which is really cool because this is completely unexpected. So actually, we think that this is a repressor of activity for wing A. Now, when I knock out the homologous region in the lady painted butterfly, again, the decorations here, we have this like a small space, and when I knock out this element that is the same that the before, is actually acting as an enhancer. So the same element that in one species is acting as a repressor, in this species is acting as an enhancer. So tinkering of these conserved regions is producing new functions in the butterflies, which is really cool. Now, monarchs are very different. We can identify mostly of the lymphatic ground plan. And what we see when we knock out the wing A gene, you notice there's this white that is spreading in the wings, and when we knock out this regulatory element, we have the same effect. But these regulatory elements, and actually every single regulatory element I have knocked out for this butterfly, it doesn't have sequence conservation with any of the nymphalids that I've seen so far. So, to summarize, we have observed that there are enhancers that are in conserved regions that are driving ancestral expression patterns, specifically around the central symmetry system. We have observed that novel functions can evolve from conserved regions. We have an enhancer that acts as a repressor in one species, as an enhancer in another species. And then we have novel expression patterns from novel sequences. With that, I want to thank the Reed Lab, the wonderful undergrads doing a lot of work preparing the lab, and also Arnaud Martin and George Washington University. Any questions?
Is any of the changes you're seeing with the CRISPR knockouts having to do with the structure of those cells? Because I know there's sometimes like the melanin layer, all that can change the way the color reflects. What are you just taking those?
It's way early earlier than,
Oh, it's earlier than that.
very early before. So a lot of the pigments that in the codcy or the structure of the scales occur during pupae, and this gene actually is doing all the pre-patterning during carotid. So it's basically removing like the signal. It's like drawing the sketch of the lines of where the patterns are going to be. So that's what is going away.